Hello students, I am teacher Svetlana and in our today's lecture we will learn about the nervous tissue, the neurons and its types, which is covered in the chapter Animal Tissues. So let's begin. The nervous tissues. Nervous tissues is composed of nerve cells or neurons and neuroganglia. So the nerve cells are also called as neurons. Neuron is the structural and functional unit of the nervous system. Have a look at this image here. This is your neuron and these are your neuroganglia, which are also called as the supporting cells. So the neuroganglial cells are non-nervous supporting cells that fill in the interneuronal space. The neuroganglial cells are capable of regeneration and division, but neurons are not capable of regeneration because of lack of centrioles. So these can regenerate, but the neurons cannot regenerate. So the spaces between the neurons is filled by these supporting cells. Intracellular matrix is absent in the neural tissues. Neuron is an impulse generating and impulse conducting unit. So these neurons, they can generate impulse and they can conduct impulse. They bring about quick communication within the body. Neurons change action potential of their membrane on receiving an external stimulus. This property of neuron is called as excitability. They also carry a wave of electric impulse from the dendron to the axon. That is the process of the neuron. So these here are the dendrites. The dendrons can carry impulse from here to the axon, which is another extension of the cell body of the neuron. Now this process is called as conductivity. Now students, let us understand the structure of a typical neuron. A neuron is made up of cytone or the cell body. Have a look at this image here. This is the cytone, which is also called as the cell body. It contains granular cytoplasm called as neuroplasm and a centrally placed nucleus. So this here is your neuroplasm and the central nucleus. Neuroplasm contains mitochondria, Golgi apparatus, endoplasmic reticulum and granules called as Nissels granules. They are made up of RNA. Cell body gives out two types of processes, namely dendron and axon. This here are your dendron and this long one is the axon. Dendrons are short branched processes. The fine branches of the dendron are called as dendrites. These here labeled the fine branches of your dendron, which is called as dendrites. They carry impulse towards the cytone. So they receive an impulse and they carry the impulse towards the cytone or the cell body of the neuron. Further, an axon is single elongated cylindrical process. Have a look at the image again. This is long single elongated cylindrical process. Axon is bounded by an exolemma. The protoplasm of axon is called as axoplasm. It contains large number of mitochondria and neurofibrins. Axon is enclosed in a fatty sheen called as myelin sheath. Now here you will see that it is enclosed in this fatty sheath. This sheath is called as myelin sheath. Outer covering of the myelin sheath is called as neurolemma. Myelin sheath and neurolemma are part of another cells called as Schwann cells. So this together forms your Schwann cell. Schwann cell shows nucleus at the periphery. Can you see the nucleus here, which is at the periphery? The myelin sheath is absent at the intervals along the axon. So at intervals you will see, can you see this blue region on the axon? So here the myelin sheath is absent and this place is called as node of Reniers. The terminal arboration of the axon is called as telodendron here. Where it terminates, this is the axonal terminals and this end is called as the telodendron. Students, based on their functions, neurons are classified into three types. We have efferent neuron and interneuron or association neurons. Let us have a look at each of these three types individually. The efferent neuron, it carries impulse from the sense organ to the central nervous system, that is your CNS. Hence, it is also called as the sensory neuron. Have a look at this image here. Can you see here it is marked as the sensory neuron? This is your efferent neuron. This is the direction of the conduction of the impulse. These are your dendrites which are in contact with your sense organs. So these carry the impulse from the sense organ towards your central nervous system. Then we have the efferent neuron. It carries impulse from the central nervous system to the effector organs. Have a look at this image here. It carries the impulse from the central nervous system to the effector organ. That's why they are also called as motor neurons. Now, efferent neurons, they are found in the dorsal root of the spinal cord, and on the other hand, the efferent neurons, they are present on the ventral root of the spinal cord. Then we have the interneuron or the association neurons. Now these are located between your sensory and motor neuron. Have a look at this image here, which is labeled as 2. This is your interneuron. You can see it is present between your sensory neuron and the motor neuron. What are the functions? These perform processing, integration of the sensory impulse, and activate appropriate motor neuron to generate motor impulses. So they process the impulse that is there here. This is your interneurons. They process the impulse, they integrate it, and they activate the appropriate motor neurons to generate the motor impulse. Furthermore, students, depending on the presence or absence of the myelin sheath, neurons are classified into 2 types, that is myelinated and non-myelinated nerve fiber. So let us have a look at them. The myelinated, which is also called as medullated nerve fiber, have a insulating fatty layer called as myelin sheath around the axon. If you remember, when you saw the structure of your axon, we saw these typical swan cells that were present, which are made up of a layer of insulating fatty layer. Now this makes the fiber appear white in color. This sheen is secreted by the swan cells. The sheen is not continuous. It is interrupted at nodes of Renivers. So it is not a continuous sheet. There is gaps in between, which are called as nodes of Reniver. Neurolema surrounds the axon. Now, the impulse is conducted at a faster rate in such a nerve fiber because it jumps from one node to the next. So here along the axon, you can see that the impulse jumps from one node to the next, and in such a way, the impulse is conducted at a very faster rate. Now, such transmission of impulse is called as saltatory conduction. Myelin sheath prevents the loss of impulse during conduction. Your cranial nerves of the vertebrates are myelinated. The swan cells of the non-medullated nerve fibers do not secrete the myelin sheath. So here this is a non-myelinated axon. The myelin sheath is not there. These fibers are grey in color due to absence of the fatty layer. Conduction of impulse in the non-myelinated fiber is slower as compared to the myelinated nerve fibers. Nerves of the autonomous nervous system are non-myelinated. Students, the functional contact between the axonal ends and the dendrites of the adjacent neurons is called as a synapse. Have a look at this image here. You can see this is your neuron, and here this tiny thing that is there, the telodendron of this axon is coming in contact with the dendrite of the next axon. So this region is highlighted here. You can see that this is the axonal end, that is the telodendroid region of this neuron, and the next below is the dendrite of the next neuron. So this joint here where they come in contact is called as the synapse. So you will learn about synapse in the chapter Control and Coordination in your class 12. That is the impulse that this neuron has received. It passes through the axon, comes to the telodendrite region, and from here it is received by the dendron of the next neuron. So this transmission of impulse you will study in detail in class 12th. Now students, based on the number of processes given out from the cyton, that is the main cell body, neurons are classified into three types. We have unipolar or monopolar neuron, bipolar neuron and multipolar neuron. You remember we have studied that there are two processes that come out. One is the axon and the other one is the dendron. So now we'll see what is a unipolar neuron. It has a single process originating from the cyton. Both the axon and the dendron arise from the cyton at one point. They conduct impulse to the central nervous system. Example, neurons of the dorsal root ganglia of the spinal nerves. So here, have a look at this. This is your unipolar neuron. So such neuron is present in the dorsal root ganglion of your spinal nerve. Then we have here, you can see it's just at one point. Then we have the bipolar neuron. It has two processes. A single dendron and an axon are given off from the opposite poles of the cyton. So have a look at this image. Here you can see this is the cell body, cyton, and in two opposite directions, two processes have come out. So this is a bipolar neuron. They bring about transmission of special senses like sight, smell, taste, hearing, etc. Neurons of the retina of your eye and the olfactory epithelium are of this type. Then we have the multipolar neuron. Cyton is star-shaped and gives out more than two processes. There is only one axon and the remaining are dendron. So have a look at this image here. This is your multipolar neuron where you have one long axon and you have more than one dendrons that have come out which split into dendrites. Axon initiates from a funnel-shaped area called as axon hillock. So this here is your funnel-shaped structure from where the axon comes. This region is called as the axon hillock. Here also in the bipolar neuron, you can see from this funnel, your axon has come out. So this is the classification based on the number of processes that are coming out from the cyton of the neuron. Thank you students for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe and keep learning.